right, I think we'll get started. Thanks everyone for coming out today for our seminar by Dr. Forrest White from MIT, where he's a professor of biological engineering and also a member of the Koch Institute for Integrative Cancer Research. So by way of brief introduction, Forrest earned his PhD 1997 in analytical mass spectrometry, working with Alan Marshall at Florida State University. He moved on to postdoctoral work with Don Hunt at the University of Virginia, which I think during this time really kindled his interest in phosphorylation signaling, and took a bit of a detour in the biotech industry for a couple years before joining the faculty at MIT in 2003, where he's been since, and his lab continues to be grounded in addressing questions around phosphorylation signaling. I'll take a minute before Forrest takes the stage to reflect a bit on the impact of his work. So in the time frame of his postdoctoral years, he was leading the development of an approach to enable broad enrichment of phosphorylated peptides from complex proteomes, and followed by quantification of specific changes in specific phosphorylation sites by mass spectrometry, what we refer to today as phosphoproteomics. And to me, the chronology is kind of interesting because during this time, you had the first drafts of the human genome coming out, and many of our colleagues were eager to use the tools coming out of the Human Genome Project to start interrogating classes of known drug targets like kinases, right? A lot of interest in kinases. So people would use these tools systematically deplete or overexpress kinases in a specific genetic context, maybe particular driving mutation or expression of oncoproteins from DNA tumor viruses, et cetera, to understand how the kinome was rewired, if you will, in the context of, for example, cancer. But what was missing at this time, so you could get these really nice genotype to phenotype relationships, disrupt a kinase, cell death, something like that. But what was missing really was kind of an unbiased way to understand how, when you perturb a kinase, how that propagated throughout the broad signaling networks in cells. So Forrest's paper that was published in 2002 during his postdoc really laid the foundation for how we tackle this problem today. So labs, all around the world have built on this sort of foundational technology, made improvements and all such to address interesting questions. And maybe a nice kind of physical representation of the impact of his work is found when you look at, for example, these databases of phosphorylation sites, Phosphocyte Plus and things like this. The vast majority of those sites have been identified using phosphoproteomic techniques. And so sort of the pendulum, I think, has swung now completely the other way, where there must be something approaching like 300,000 phosphorylation sites in these databases. And maybe only about 10,000 of those have been convincingly assigned to the activity of a specific kinase. And virtually all of these reports can draw a straight line to Forrest's original paper. I think that's really great impact. So over the years, Forrest Lab continues to be at the forefront of using these technologies in phosphoproteomics to address questions in basic biology, translational, and clinical research. And judging by the title of his talk today, I think we're going to hear latest results from sort of the translational and clinical end of the spectrum. And before I turn the floor over to Forrest, I just want to tell you how hard this intro is for me, because I've known Forrest for like 30 years now. And so I desperately want to sit up here and just bust on him. But I want to recognize the great venue we have for seminars in oncology. So welcome, Forrest. The floor is yours.
I was waiting with trepidation for where that was going. That was nicer than anything I expected, which is cool. All right, so I'm going to talk about some of the work we've been doing over the last few years in the phosphoproteomics realm, and really focusing on one aspect of phosphoproteomics, which is not necessarily super well represented in these large databases, but is phosphotyrosine proteomics. And our focus throughout the talk today is actually going to be on pretty much solely phosphotyrosine proteomics. And so why do we do that? Well, serine phosphorylation is like 90% of the phosphoproteome, and threonine is 10% of the phosphoproteome. And then phosphotyrosine basically makes up somewhere between about 0.1 to 1% of the phosphoproteome. So it's really low level, but it's also very highly regulated. And as most of you know, most oncogenic drivers are aberrantly activated kinases, and most of those are actually aberrantly activated tyrosine kinases. And most targeted therapeutics, basically for cancer, target activated kinases. And again, most of those are targeting tyrosine kinases. And so for really understanding, especially in the field of cancer, what are relevant targets, we can read out a lot of that from tyrosine phosphorylation. And in fact, as we look at, for instance, the pathways that are driving cellular proliferation, and this is sort of downstream of EGFR and ERB family members, every place where there's a red circle, there's a protein that is tyrosine phosphorylated, where that function of that tyrosine is relevant for promoting that proliferation signal. And similarly for migration, when we look at migration or invasion, every place where there's, in this case, it's a lightning bolt rather than a red circle, but you get the idea. Every place where there's a lightning bolt, there's a protein whose activity in this network is regulated by tyrosine phosphorylation. And so by reading out tyrosine phosphorylation, we can read out proliferation, we can read out migration. In work that we're just getting ready to publish now, we can read out cell metabolic activity actually is somewhat regulated by tyrosine phosphorylation. And so if we can study this, then not only can we identify some of these sort of classic behaviors of oncogenic cells, but we can also identify drug targets. And this really nice review by Pasayane's lab looking at the timeline of clinical success for kinase inhibitors in cancer. Every place where there's a pink box is a tyrosine kinase inhibitor that's been approved in the clinic. You can see that there's a lot of those over the last 20 years. The blue boxes are serine threonine kinase inhibitors, largely in the RAS-MAP kinase pathway. And then the gray boxes are basically the PI3 kinase inhibitors. And all of these can basically be detected by tyrosine phosphoproteomics because the activation loop of these kinases tends to be regulated by a tyrosine phosphorylation site. For the tyrosine kinases themselves, of course, we can read out the activation loop for these kinases, but more importantly, we also read out the substrates. So we can start identifying activated kinase signatures in various patient tumors and therefore start identifying which inhibitors they might respond to. And that's really going to be the subject for most of the talk today is trying to use this information and translate it to the clinic to identify the right targets and the right treatments for each patient. All right, and so that's the backdrop to the talk, which is really trying to improve clinical diagnosis and patient stratification by directly identifying activated pathways regulating these critical processes in cancer. And the other way that people have tried to do this, as Jared was mentioning, is through genomics or transcriptomics. So you could basically take each patient's tumor and then read out the genetic mutations that are there, or read out the transcripts by RNA-seq or single-cell RNA-seq, however you want to do it, read out the transcripts that are there, and then use that information to try and infer the activated kinases that are present. But there's that critical word of infer, right? You're trying to say that because I see these changes in the transcriptome, or because there's this mutation, I think this kinase is going to be active, and therefore it's a good drug target. For instance, in lung cancer, they look for EGFR mutations, thinking that that might be indicative of which patients are going to respond to an EGFR inhibitor, but it's missing all the other stuff that might be there. It's missing as I'll show you, it's missing some of the critical resistance pathways that are known to be regulated and might regulate patient response. But it's also missing this nuance of whether or not that activity is even there in those cells in that patient tumor. And our goal here is really to use phosphotyrosine proteomics to directly measure activity of these pathways and to do this in a clinically relevant setting. And so the challenge really is really here that the phosphotyrosine phosphoproteomics is really an art form. And data quantity and quality varies from lab to lab, and even within my lab varies from user to user. And so that by default sounds like it's not ready for clinical prime time yet, because if it varies from user to user, well, how are you going to get CLIA approval for something that may or may not read out the same each time it's run? And so we really need to get past that and get this ready for clinical diagnostics. And I'll show you sort of our plan to do that, and how we're hoping to actually move this forward to real patient care in real time. All right, so the particular disease I was going to focus on today, my lab tends to work on several different types of cancer, but one of them that we've been focused on lately is triple-negative breast cancer. And I think most of you already know, I've talking to the Dana-Farber Cancer Center, it feels like I'm, anyway, preaching to the choir about this, but breast cancer affects a lot of women. Triple-negative breast cancer is the most aggressive form of breast cancer with the worst diagnosis. For the women with triple-negative breast cancer, about 50% or so of them are going to have a response to chemotherapy. The other 50% are going to have an incomplete response to chemotherapy, where some of the tumor will be remaining at the end of the chemotherapy cycle. For those women who have incomplete response, they're considered as chemotherapy resistant, and treatment options for those women are minimally effective. At that point, basically, most of those women are going to fail and end up dying from the disease. And so I met this team at the Mayo Clinic in Rochester that's led by Matt Getz. And Matt and I met in Rochester, and then he came to Cambridge, and we chatted some more. And I asked him sort of what was the hardest problem that they have, and where could we have the biggest impact. And he said really on working on identifying new therapeutic targets for chemotherapy-resistant triple-negative breast cancer. And so that's the project that we started working on with Matt and Judy and Liwei, the team at the breast cancer team at the Mayo Clinic. And then more recently, we started working with Natalie Kravchenko-Balasha at Hebrew University in Jerusalem. And as I'll show you, Natalie does the bioinformatic and computational modeling of our data to try and identify therapeutic targets on a patient-by-patient basis. And then Matt, Judy, and Liwei are the critical clinical collaborators that provide us with sort of the real-world samples. Okay, so how do we do this analysis? In this project, we basically started with about 80 triple-negative breast cancer PDX tumors. About a third or half of them are chemotherapy resistant, and the other are chemotherapy sensitive, so we can compare some of the signaling networks between the sensitive and resistant triple-negative tumors. Each of those are grown as patient-derived xenografts in the mammary fat pad. The tumors are grown to a couple hundred millimeters cubed, and then they're resected and frozen and then shipped to us for our analysis. And each one of those tumors ends up being processed to peptides. Those peptides are digested and then desalted, and then TMT labeled so that we can basically do multiplex analysis for quantification. We can figure out which peptides are coming from which patient tumor. And then we go in and we pull out the phosphotyrosine peptide specifically with an anti-phosphotyrosine immunoprecipitation. That's not as specific as we want, so we do a second step of enrichment where we basically get rid of any non-phosphopeptides, and we use this immobilized metal affinity chromatography for that. At the end of that, we get enriched phosphotyrosine peptides that are effectively bar-coded, so we know how much of each phosphotyrosine peptide came from each of the different tumor specimens. So we get relative peptide quantification and identification of the phosphorylation sites. All right, so what does the data look like? This is a signaling network that's sort of enriched in one of the chemotherapy-sensitive tumors, and then a portion of a signaling network that's enriched in a chemotherapy-resistant tumor, where there's actually a lot more signaling in that tumor than in some of the other tumors in the cohort. So this is nice, and we can see some of the sort of string-based protein-protein interaction networks that are present in these tumors, but it doesn't necessarily highlight the drug targets. And as we're looking across the different PDX samples, a few of which are shown here, one of the things you can appreciate maybe is that if we look at, these are PDXs from different, so the same PDX grown in different mice, and there's some variability from mouse to mouse. There's inter-mouse variability, as there always is, as we're looking at some of these biological samples. But each of the PDXs is sort of clustering within itself. So PDX2 clusters with itself, even though two of the tumors are from different mice and look slightly different. one of the things we're missing here, which when we started doing some clinical work many years ago, some analysis of glioblastoma specimens, I thought there'd be this like dark red line that just went all the way across that was like the drug target for this disease. Turns out I was incredibly naive as usual. And we don't see a dark red line that goes all the way across because, of course, the drug target for any one of these women is going to be different than the drug target for other women because their signaling networks are different, right? And what we're reading out here are sort of the signaling networks, where in one case we might see a dark red patch for patient number 2, but you can see that there's really not that same dark red patch going across the other patients because that target might not be there for all of the patients. It really needs to be at a personalized level. So given the complexity of the data, how do we then figure out which targets are best for which patients? One of the ways that we're doing that is with this information theory-based approach. And this is something that Natalie developed when she was a postdoc in Jim Heath's lab back when he was at Caltech. And they called it, at the time, surprisal analysis. And I always make the joke that it works surprisingly well. But it does. It actually works surprisingly well at identifying signaling networks and the central nodes within the data. And it's based on information theory, and what they are trying to identify using a mix of a priori knowledge coupled with the quantitative intensity of the signaling from our quantitative intensity of the phosphorylation sites from our data is to identify what they call unbalanced processes. These signaling networks that are upregulated in a given patient's tumor compared to some of the other tumors that are there, and what it might be common in a few different tumors and not in others, but really trying to identify the networks that are there as well as the central nodes that might be the good drug targets. And so they've changed this now, instead of calling it surprisal analysis, since we're doing signaling network analysis, they call it PASS, our patient-specific signaling signature analysis. It's like a tongue twister. So this PASS analysis, we can apply to this collection of PDX, a collection of phosphoproteomics data from our PDX tumors, and then start identifying the networks and central nodes that are effectively good drug targets. And in this case, what we see is that in BR98, one of the PDX tumors, EGFR ends up being one of the central nodes that's regulating BR98. And it ends up being like the node that's regulating BR98. And so the prediction is if we treat with an EGFR inhibitor basically by itself, we should have an impact on this tumor. And it works pretty well. So we treat with erlotinib as an EGFR inhibitor and compare that to Taxol, our control, and you can see that erlotinib actually controls tumor growth pretty well by itself. At the end, at 30 days or so, we start to see this outgrowth of tumor, probably because the tumors are starting to develop some sort of resistance to this single therapy treatment, which is commonly seen. On the other hand, if we look at PDX11, what we see is not EGFR by itself, but EGFR and IGF-1R networks that are both activated in this tumor. And the prediction, therefore, is that EGFR inhibition by itself isn't going to work very well because we have IGF-1R that's already active in this tumor, and that we're going to have to hit both of these if we want to have a strong effect. And you can see, looking at these lines, that basically erlotinib by itself, that yellow line, the tumor is already growing out with erlotinib alone. If we use AG1024 as the IGF-1R inhibitor alone, it doesn't work very well either. The tumor is growing out if we just inhibit that network. If we use EGFR inhibitor erlotinib plus trametinib to hit MEK in the RAS-MAP kinase pathway, trametinib is a super potent drug, and so we get some knockdown of tumor growth there. But really, when we treat with the predicted EGFR plus IGF-1R inhibitor in that green line, what you can clearly see is that we can flatline this tumor growth as long as the drug is there. When we take the drug off, the tumor starts growing out, not surprisingly, because the tumor's still there. It's not like we've wiped out the tumor. Okay, so one of the things that emerged from our data is that Src family kinase signaling was present in many of the chemotherapy-resistant tumors. It was also present in some of the chemotherapy-sensitive tumors. So it's not like this is driving by itself chemotherapy resistance, but was enriched in the chemotherapy resistant versus chemotherapy sensitive. And so we wanted to see what would happen if we would hit just the Stark family kinase network. And as most of you might know, SFK inhibitors like dasatinib or saracatinib or bosutinib have generally failed in the clinic as single therapy agents, except for chronic myeloid leukemia. But for solid tumors, they've generally failed as single therapy agents. And so we wanted to just see whether this was true in some of these PDXs. And what was surprising to us is that when we started treating with dasatinib, for instance, in this PDX that had a very high Stark family kinase signature, we could basically flatline the tumor for as long as the drug was there and for another 35 days after. And when we first generated this data and showed it to Matt Getz at the Mayo Clinic, he said this is the best effing data he's ever seen, because trying to flatline the tumor growth for these triple negative breast cancers, which are generally pretty aggressive, ends up being really challenging. And nicely, as we started looking at across other PDXs, we could basically, using our phosphoproteomics data, predict whether or not the PDX was going to respond to this SFK inhibitor by looking at the intensity of this SFK signature. So when we had high SFK signatures, like we see on the left-hand side of the screen here, we could basically flatline those tumors. If we had a low SFK signature, the tumor would basically grow out during treatment, as you can see on the right-hand side of the screen here. And so this, based on phosphoproteomics, we could read out with actually pretty nice predictive power how these tumors were going to respond to SFK inhibitor treatment. And so the challenging thing is, dasatinib's already failed in the clinic twice for triple-negative breast cancers. So that raises the critical question of how physiologically relevant are these PDX tumors? They're tumors growing in a mouse. They're not human tumors growing in a patient. So are these signals that we're seeing common to human tumors, right? So to answer that, we need to be able to access human tumor specimens. And then the other question that goes with that is, how common is this SFK high signature in triple-negative breast cancer patients? If it's in 10% of patients, maybe we could actually resurrect this drug if we had a good test for it to stratify patients onto this drug. We could have a drug that would impact 10% of patients, which would be fantastic. I mean, it wouldn't be a cure for triple-negative breast cancer, but even if we were able to impact 10% of patients and help them with as a therapeutic option, it would be great. But the challenging thing, of course, to answer both of these questions, or all three of these questions, is that we need to be able to access human tumor specimens. And to do that, we really needed to be able to perform phosphotyrosine analysis on FFPE tissues directly from human patients. And at the time when we got to this stage, we had actually gone back to the transcriptomic data and done RNA-seq on each of these PDXs, and then tried to identify the signature for Stark family kinase activation in those PDXs. We treated many of them with dasatinib or without dasatinib to see if we changed the signaling of the Stark family kinase, could we see that transcriptionally? Because, of course, if we could find a biomarker for this at the transcription level, it'd be easy to move it into the clinic. But so far, at least, SFK activation seems to be completely invisible by RNA-seq based measurements. And so we were really stuck with trying to do this using phosphoproteomics, but that meant we had to develop a new technology for doing phosphoproteomics from FFPE. And so luckily, I had this fantastic student in the lab, Ishwar Kohel, who is a former MIT undergrad who decided to stay at MIT for his grad work and join my lab. And I told Ishwar we needed to be able to do phosphotyrosine from FFPE. And so we got these tissue specimens from my constant collaborator, Jan Sarkari at the Mayo Clinic, where they had sort of hundreds, if not thousands, of PDX FFPE specimens for glioblastoma. And so Jan's technician put a bunch of them in a bag and sent them to us. And Ishwar comes into my office and he just takes this FFPE block out of the bag and puts it on my desk and he goes, "So I got these." And I'm like, "Yeah." And he goes, "What do I do?" And I'm a very bad advisor, I'll just say, because I'm like, how the hell should I know? You're the grad student. I mean, I think this is your job. Like, you figure it out, and then you come back and give me the results, and then I claim credit. Anyway, so Ishwar he sort of just shakes his head and puts it back in the bag and leaves my office, realizing that, like most people do, that I'm totally useless. And like 2 weeks later, he comes back and he's got data. And because he's like basically dove into literature. Some people had done phosphoproteomics done proteomics on FFPE. He copied some of their methods and then figured out that it wasn't quite working, so he optimized it. And within a couple weeks, he had data. And he had data not just off of the block, which would have been impressive, but he had data off of one 10-micron section off of each block, which was actually super impressive because the amount of tissue that we needed for this, what we started realizing is the amount of tissue we needed was fairly minimal. And the nice thing about that is we can go to the clinicians and say, we need a single 10-micron section off of your block to figure out what's happening with this patient. And they're always willing to do that, basically, because that's almost nothing for them. So as long as there's any of the block remaining, we can basically get tissue for almost any disease where they've gathered FFPE tumors in their tumor archive. Anyway, so the peptide yield is linear with tissue amount. We can quantify hundreds of phosphorylation sites from as little as one 10-micron section. It covers the tyrosine kinome branch of the kinase tree pretty well as some of the activation sites. So what does it look like for triple-negative breast cancer tumor specimens? At the Mayo Clinic, they had taken 10 or so patients, and when they resected the tumor, they took part of it and froze it in liquid nitrogen, and the other part of it they put into FFPE. So we had a direct comparison of sort of the gold standard, which is flash frozen tumor specimens, compared to FFPE preserved tumor specimens. And then using our technology as we had it at the time, we compared those two, and the data shown here, each of the tumor specimens from each patient tends to cluster together, which is good. They're not quite identical because they're different sections of the tumor, for one thing, so there's spatial heterogeneity there, but also there is some artifact of the fixing time, and we've studied this and published on it, that if you're fixing a larger tumor, it actually, you can have some artifact on the signaling networks. But if you're fixing a biopsy, for instance, that fixing happens almost instantaneously, and therefore there's actually much less artifact on small tissue sections. Anyway, from the data, either frozen or fixed, we could start reading out the active signaling networks in each patient's tumor just by looking at the phosphoproteomics data. And this is without any fancy informatics. We basically just look at the signaling networks that are there and the intensity of signaling. We can read out for patient 1, for instance, there's really high levels of EGFR signaling and almost nothing else. So that patient we would predict might actually respond to an EGFR inhibitor. Patient 2 has EGFR signaling, but it also has very high levels of MET. MET is a known resistance pathway for EGFR. So regardless of EGFR mutation status, if you have MET signaling, the patient's probably not going to respond to EGFR inhibitor alone. And therefore, this patient would probably most likely be treatable maybe with an EGFR inhibitor and a MET inhibitor combined, or some bispecific antibody. Of the 10 patients we analyzed initially, one of them had high Sarc family kinase signaling. So this sort of type of data would suggest that maybe there's a small percentage of patients that have high SFK signaling. However, this is only 10 patients. So with the Mayo Clinic, we've recently finished the analysis of 150 triple-negative breast cancer patient specimens. Each of these patients, we had one 10-micron section off of their block, and we're able to quantify in total around 3,000 tyrosine phosphorylation sites. We can start identifying and clustering the patients based on their signaling, and you can see that there's a few different clusters of patients that are revealed by doing co-correlation plots of the patient signaling data. Turns out when we just look at the intersection of signaling across all 150 patients, what we see is that there's sort of 3 categories of patients that emerge from this data. The first one is really driven by tyrosine phosphorylation on RNA processing and protein translation and co-translation machinery. What's kind of interesting about this is that these are tyrosine phosphorylation sites on many of the splicing factors that are involved in RNA processing and RNA binding proteins. These are unknown and poorly characterized tyrosine phosphorylation sites. We've been doing tyrosine phosphorylation for the better part of 20 years, and we've actually never seen these sites before. but suddenly they're there and actually fairly high on about a third of these triple-negative breast cancer patients. And there's really no other driver present in these patient tumors, which is a little bit disturbing because we don't know how to treat this yet, but I think this is a fascinating area of biology to try and understand what's happening when we get tyrosine phosphorylation on these sort of RNA processing machinery. The middle part of this is driven by immune signaling, and that's really the strongest signature that comes out. And then the bottom third or 40% of the patients really have either RTK or Src family kinase signaling. For the middle third, what we see is that these patient tumors can really be stratified into immune hot or immune cold tumors just based on the phosphotyrosine data. And instead of looking at the amount of TILs, we also have TIL data for each of these patients, so we can correlate that data with our phosphotyrosine data. But instead of looking at the number of lymphocytes that are present in each of these tumors or TILs present in each of these tumors, we can read out the signaling network that's there for activated T-cell signaling. And instead of, again, just one phosphorylation site, we get multiple phosphorylation sites on the ITAMs, the active tyrosine phosphorylation sites on the TCR complex. And we also get some of the signaling networks downstream of that TCR complex that suggests that these tumors actually have T cells in there that are actively signaling. And for patients that might respond to checkpoint inhibitors, we could start trying to stratify patients based on whether or not they have active T cells in their tumors. And for triple-negative breast cancer, the response rate ends up being about 20 to 30% of those patients might respond to checkpoint inhibitors. Our data would suggest that for this 150 patient cohort, we get about 30% of those patients that have this active T cell signaling in their tumor. We still have to do the exact study to actually look at patients who have gotten checkpoint inhibitor and see whether we can predict their response, and that's sort of what we're moving toward now, because I think that'll be critical as we try and move this into the clinic to actually get that clinical response data. The other thing we can do is basically look at the signaling and identify RTK activation points, RTK activation pathways, and this will point to sort of targeted therapeutic options. And you can see that, for instance, one of these patients has really high signaling by MET and could be responsive to a MET inhibitor. Other patients have high signaling by FGFR or VEGFR or EGFR. And so we can start trying to identify which therapeutics might be effective for which patients based on our phosphotyrosine data. Okay, so that's using our sort of standard protocol so far, and again, in a not so clinically relevant assay, because each person in my lab gets slightly different results, and therefore, and slightly different numbers of phosphorylation sites, et cetera, and therefore, trying to make this into a CLIA-approved platform would be really challenging. But to do that, we can turn to an approach that we've co-developed with Thermo Fisher Scientific called SurQuant phosphotyrosine. And the idea behind this approach is that for each one of the phosphorylation sites that we're interested in, and in this first assay, we had 380 or so tyrosine phosphorylation sites that we identified from clinical tissue specimens that we thought were relevant for identifying the right therapeutic targets for each patient. And we took these 380 tyrosine phosphorylation sites and made heavy labeled peptides for each one of these, heavy labeled phosphopeptides for each one of these. And then we can take those heavy labeled phosphopeptides and we can spike them into our samples as soon as we basically do this tryptic digest, we can spike them into our samples and then use those as internal triggers to basically trigger the acquisition of the endogenous peptide. And now we have an internal standard for each of our peptides of interest. So we spike in those heavy peptides, we do our IP that pulls down the heavy and the light, we do our second stage of enrichment, and then we throw it into our mass spec. And the way that the mass spec works is by looking for the mass to charge ratio of that heavy peptide. When it sees it, it does an MS-MS, basically it fragments it and looks at the fragment ions. If those fragment ions match the predicted fragment ions that we expect would be there for that peptide, then it starts going back and forth between the light and the heavy and gathering fragment information from the light and the heavy during the chromatographic elution profile. And that's shown here on the lower right-hand side of the screen. And then we can start basically quantifying the light to heavy ratio during the elution time. And that basically gives us an internal standard that we're comparing against. So this would be similar to what's done in the clinic for instance, Olympic testing, where they're doing multiple reaction monitoring by mass spec to look at doping of Olympic athletes. It's exactly the same technology, exactly the same approach, but now using this to do that same thing for hundreds of tyrosine phosphorylation sites so that we can read out in a clinically approved type of format which signaling networks are present for each patient. And just one more quick note on this. The other thing that we've done is to, and I don't have slides on it today, but is to do this with multiple isotopologs, so multiple heavy isotope labeled peptides per site, if you will, or per peptide, endogenous peptide. And that gives us internal standards that we can then, and an internal standard curve that we can then regress against. So it's no longer a one-point calibration curve, but instead a multi-point calibration curve for each of our peptides, giving us true analytical accuracy across these different specimens. All right, so this was 340 tyrosine sites, and then we applied this Schur-Quant phosphotyrosine, what we called 1.0, because we were going to do 2.0. That failed horribly, so we're just stuck with 1.0. But anyway, I could tell you that story later. But anyway, the Schur-Quant 1.0, which was 340 tyrosine sites, and we used that to quantify 31 signaling networks in 31 colorectal tumors. For EGFR, for instance, this is the phosphotyrosine 1197 or 1173 if you're an EGFR signaling person. And you can see that's quantified across these different patient tumor specimens where tumor 19 has a lot of EGFR signaling and the tumor next to it, tumor 21, has very little. If we start looking at the data across these 31 different tumors, what you can see is that each of the tumors looks like it's its own thing. There's a lot of patient-to-patient heterogeneity here. Again, no smoking gun that goes across all of these tumors because each tumor has a different drug target. But by identifying some of these clusters of phosphorylation sites that emerge, we can start identifying, for instance, the innate immune peptides that are regulated by macrophage signaling, and those are high in selected patients and low in many others. We can use this same sort of data, and similarly to what I showed you for our sort of data-dependent analysis, using our Schur-Quant data with more accurate quantification, we can get phosphotyrosine signaling signatures for each of these tumors. We can identify which patients have which tumors, which pathways activated, and then for each one of these, we can go in and interrogate the data. So we get a couple of patients that have high ERB signaling. If we go in and interrogate that data, the top patient has very high EGFR and very low HER2 and HER3. Again, this patient might be susceptible to an EGFR inhibitor by itself. But if we look at the patient on the bottom half of the screen, they have EGFR phosphorylation, but they also have HER2 and HER3 phosphorylation, a known resistance pathway to EGFR. Treating this patient with an EGFR inhibitor it's bound to fail. They already have the resistance mechanisms present and active in their tumor. So using phosphotyrosine, we can read out this level of data. Again, out of these 31 patients, we get 2 that have very high signaling on the tyrosine phosphorylation network downstream of the TCR, the T cell receptor, suggesting that 2 of these patients have quite a bit of active T cells in their tumor and might be actually pretty responsive to checkpoint inhibitors. All right, so what I wanted to close with are a few slides on a new center that we've just launched at MIT in collaboration with the Dana-Farber Cancer Institute here. And this is funded by the NCI CSBC, our Computational Systems Biology Consortium. And it's the longest center name ever. We were trying to max out the allowable space on our NIH grant. But it's the Center for Quantitative Systems Biology of Glioblastoma cells and their interactions with the neuronal and immunological milieu. So we were trying to get a few more words in there, but anyway, we should have put synergy and nano somewhere in there. But anyway, the general idea is that we really want to investigate the tumor neuron interactions for GBM as well as the tumor immune interactions and get a more holistic picture of the GBM tumor and microenvironment. The people on this grant, just quickly, are myself and Francisca Mejor here at the Dana-Farber Cancer Institute. We're co-PIs. And then Natalie Agar at Brigham and Women's Hospital and Dana-Farber is also a co-investigator on the grant, as is Stephanie Spranger, an immunologist at MIT. And then we have several people at the Mayo Clinic, including Jan Sarkaria, who's the lead PI there, and Antonio Ivaroni at the University of Miami. And what we're working on is really, as a team project, is really trying to use these MRI-guided biopsies and multi-omic analysis to understand the tumor and tumor microenvironment in distinct regions of the tumor. And so at the Mayo Clinic, they were doing this clinical trial, clinical trial actually, but doing this clinical trial where they were going in and doing multiple biopsies, MRI-guided biopsies for each patient that came in, and then taking cores, or taking punch biopsies or core biopsies from the tumor-rich region, so that's core B, which is contrast positive and flare positive. The invading sort of edge of the tumor, which is contrast negative and flare positive. The far edge of that, which is contrast negative and flare negative, which is going to be predominantly normal brain, but with some small amount of tumor cells. And then the last core is sort of the necrotic core of that tumor, where they go in and do a punch there. And for each one of these punch biopsies, we've been applying this multi-omic analysis, where we take that punch biopsy and we basically Z-stack it, if you will, where it's centered on the single nuclei RNA-seq, and we take a 20-micron section and apply single nuclei RNA-seq from that 20-micron section. We take the next 20-micron section up or below. We call that either pre or post, because pre is above and post is below. And we apply phosphoproteomics and proteomics to that 20-micron section. We do spatial metabolomics in Natalie Agar's lab with the 10-micron section above that. And with the section above that, we do H&E imaging. And then we do that sort of, again, pre and post that single nuclei RNA-seq chunk. And then we go down a couple hundred microns lower in that punch, and we repeat that whole process so that we get some idea of the spatial heterogeneity as we go down that punch biopsy. But we have all this data that we can start tying together to try and understand what's happening in each part of this tumor. From the H&E data images, we can see that core B is, like I said, contrast positive, flare positive. That's majority tumor cells. Core C has some infiltration of tumor cells. Core D is largely uninvolved cortex, but with some tumor cells there, although very few. And then core E is that sort of, there's some viable tumor cells there, but it's that necrotic core. So it's, there's a lot of other stuff happening in that core. So, and this is an EGFR amplified patient. So looking at our phosphoproteomics and protein expression profiling, we get on the order of 1,400 tyrosine sites, some 10,000 serine threonine sites, and 4,000 or so proteins quantified for each one of these cores. You can see the cores themselves actually cluster tightly, which is kind of cool. So whether or not we're above or below that single nuclei RNA-seq chunk, or whether we're 200 microns down in the core, it looks about the same, so it says that the spatial heterogeneity within that core is actually not that much, and therefore we can tie a lot of these omics data together because it's really the same cell biology, the same tumor biology we're looking at as we're doing each of these different omic analyses. All right, so we can look at the single nuclei RNA-seq data, and that reads out the malignant cells, which are in yellow, and then we get all these other cell types, and we can start digging through and figuring out which cell types are present in which of these different regions of the tumor, either the B, C, D, or E regions of the tumor. And that's done by Antonio's lab down at Miami. In Natalie Agar's lab here at BWH and DFCI, they've been doing spatial metabolomics, and you can see that the metabolites are actually quite a bit different and this might not be surprising for anybody who's thought about this, but the metabolites are quite a bit different as we look at the tumor versus the normal brain. There's significant different utilization and generation of different metabolites. as we go from sort of tumor-rich out to normal brain or into that core biopsy, or into that necrotic core. One of the things we've been doing now is to couple our proteomics data and our single nuclei RNA-seq data to inform each other. And one of the ways that we've been doing that is something called an approach called CyberSort-X. And so we can take our proteomics data, and then load it into CyberSort-X. And CyberSort was originally generated to take bulk RNA-seq data and back out the cell type contributions that led to that bulk RNA-seq data. You can do the same thing with bulk proteomics data and back out the cell type contributions that led to that bulk proteomic data. And the really cool thing is just using our proteomics data, we get data that's almost identical to the single nuclei RNA-seq data, but now using protein expression profiles rather than transcripts, and with different data for each, so we can start putting that data together. On the signaling side, the proteomic data basically matches to the transcript data. So EGFR positive patient, we can use EGFR as a marker of tumor cellularity, if you will. And you can see that as we look at core B, C, D, or E, we get quite a bit of EGFR protein expression where there's a lot of tumor in core B. Out at the far edge, core D, where there's very few tumor cells, we can still see EGFR, but very little EGFR is present there. And by single nuclei RNA-seq, we get virtually identical data looking at not EGFR here, but looking at actually using infer CNV to identify which are tumor cells and which are non-tumor cells. And so our data basically matches up really well. But what I think is actually really cool is looking at the difference between the protein expression and the signaling data. And so when we look at EGFR protein expression, you can see that, like I just showed you, core B, core C, core D, we're seeing less and less of the tumor cells. But when we look at the signaling data, we see that for those very few tumor cells that are present in that very far region of the punch, where there's only a few tumor cells there, they're actually signaling quite a bit. The phosphoproteomics are actually quite high. Phosphorylation of EGFR is quite high compared to the number of cells that have, or the amount of EGFR that's there. Whereas in core E, which is that necrotic center of the tumor, there's a lot of EGFR there, but there's not much signaling when we compare the protein expression to the phosphorylation. So we're starting to actually learn something about what's happening. We can take this data, and the serine threonine data in this case, and put it into the new kinase library tool that was generated by Luke Cantley's lab. And what we see here is that when we look at core B, for instance, which is majority tumor, what we get out are actually a lot of CDKs. These cyclin-dependent kinases that regulate cell proliferation are really strong when there's a lot of tumor cells there. Whereas on the other hand, when we look at core D, which is predominantly normal brain here on the edge, what we see are a lot of these GRKs, or G protein coupled receptor kinases. These are known to be activated and signaling in the normal brain predominantly. And so it's actually pretty cool because the data makes sense. I mean, sorry. But it's always impressive when we get data and then we do some analysis on it and then you get information out, you're like, oh, that's so cool. Anyway, the last thing I'll close on here is that we've been trying to integrate data now. So we took the metabolomics data and the proteomics data, and we put it together in what's known as MOFA, or this multi-omic factor analysis algorithm. And this is basically doing regression or factor analysis on multiple data types at a time. And we were looking for particular factors that differentiated the cores from each other. And factor 2, for instance, differentiates core E, that necrotic core, from sections B, C, and D, which are basically other parts of the tumor. So then we started looking into what are the things that are weighted heavily in this factor 2, and one of the things that came out was the AMPA receptor. And the AMPA receptors are known to be active on normal neurons, typically, and it was kind of surprising to us that we would see this coming out as being differential in the necrotic core. But what we see when we look at the single nuclei RNA-seq data that was not included in MOFA is we see this GRIA1, which is the AMPA receptor expression, In the normal brain, it's predominantly in neurons. You can see that in the bright green in core D on the third bar over. But in the necrotic core, now that AMPA receptor is being expressed by malignant cells. And so this leads to that tumor-neuron interaction that Michel Mange's lab has really nicely highlighted and done a lot of nice work on. And here what we're seeing is, again, those tumor cells have upregulated this AMPA receptor, allowing them now to communicate more effectively and receive a lot more signals from the neurons in this necrotic core where they have much less in the way of other nutrients and signals coming into them. All right, so what I've tried to show you today is that using our phosphotyrosine analysis, we can highlight and identify activated signaling networks in tumors. We can do this on FFPE tissues. We can also do it on biopsies. We don't need very much in the way of tissue in order to do this. Using SureQuant-based targeted analysis for basically MRM-style analysis, we can get deep coverage and retain accurate quantification using internal standards, which is critical if we want to move this into the clinic. And what we're hoping to do is use this approach to do patient stratification to improve therapeutic responses. And then lastly, we can do multi-omic analysis and get some unique insight. So the people who did all the work I showed you today are shown here. Ishwar Kohale developed our FFPE strategy. Lauren Stopper and Cam Flower developed our SureQuant phosphotyrosine approach. Tigges did the analysis of the 150 breast cancer tumor specimens. And Yajin has been working on and has done all of the work on that the U54 project I showed you at the end where we did the spatially guided biopsies. And that was done in collaboration with people at the Mayo Clinic, people at Thermo Fisher, Natalie Kravchenko and Kravchenko-Balasha. And then I'll finish with our project personnel again because this team is allowing us now to start digging deep into these tumor specimens. Okay.